All right, well, thank you very much. Really exciting to participate here. So I lead the AI for Science lab at Stanford. And one of the ideas that I'm really excited about recently is thinking about how do we try to scale the number of agents, the number of AI scientist agents that actually collaborate and work together. So I think that's actually a really exciting new direction of scaling. So instead of scaling individual models, but scale the number of agents, from tens of agents to hundreds to thousands and even millions of agents that collaborate and see what are the new kinds of capabilities that emerges. So I want to give some examples of that kinds of agentic scaling. So the first step we took in this direction is we created this virtual lab, which sort of has like initially a team of 5 to 10 agents that sort of emulate my physical lab at Stanford. So in the virtual lab, we have an AI professor agent that runs the lab. And then working with the professor, we have a bunch of different AI students with different expertise ranging from mathematics to data science to protein design. And the agents have their own group meetings. We give them a budget so they can actually run some experiments. We also created for the agents a replica of Stanford agent school where they can go to the school to become better researchers in their specific domains. And after we created this virtual lab, we give the agents several different tasks. So here in this demo, I'll show you sort of the first project we assigned to the agents, which is actually help us to design good binders to some of the recent SARS-CoV variants. Right, so in this demo, right, so this is sort of illustrates one of the agent group meetings where the professor first explains to the student agents what is the goal here of the project, and then the different agents, different experts then can speak up and contribute their ideas. Right, in this case, the immunologist recommended designing a nanobody, and then the machine learning agent then come up with some ideas how to optimize these nanobodies. So after a series of these agent group meetings and one-on-one discussions between the agents, right, they actually designed a pretty interesting computational framework for optimizing these nanobody proteins, which is different from what people have published before. And then by running that framework, then they came back to us with a few of these new nanobodies that they designed. And then we, with our experimental collaborators, then we synthesized these proteins and we tested them in the real world to show they were actually quite effective binders. In particular, they were actually better binders than the best previous human-designed nanobodies to some of the recent COVID variants. So this to us was actually quite an exciting first step to illustrate now how a team of agents, when they collaborate, can actually solve these quite challenging open-ended projects that we can then experimentally validate. So for our next step, where we want to be even more ambitious, right? So we said, okay, so can we actually now have try to emulate a much more complex organization of like an entire pharma company now with tens of thousands of AI scientist agents. So we call this the virtual biotech. So in the virtual biotech, we have a CSO agent, the chief scientist officer agent that runs the virtual biotech with its own chief of staff, its own office and lawyers and so on. And then under the CSO agent are these different divisions that sort of mirrors the standard divisions you see in the human pharma company. For example, we have a division of agents that looks for good targets, another division that designs the molecules given those targets, a third one that designs clinical trials and looks at safety and so on. And all of these divisions now are operationalized by different AI scientist agents with different roles and specializations. For example, under the target identification division, right, there's one agent that's really good at looking at all the genetics data, another agent that's really good at doing single cell analysis and so on. And all of these agents then can collaborate and tackle these much harder problems. So here's one example of the project we assigned to this virtual biotech now, which is to help us to design new therapeutics for a particular target in lung cancer called CD276, which is like a transmembrane protein that has the effect of suppressing immune response. Right, so there's a lot of pharma companies that are interested in this target. So we said, okay, can the virtual biotech actually help us to design a new therapeutic for this target? And then this team of agents, involving many thousands of agents, then they actually did a lot of comprehensive analysis, looking at both genetics data, genomics data, patients data, and so on. And by analyzing these disease mechanisms, then they came back to us, right, with actually a specific design, what's called an ADC or antibody drug conjugate, where you have an antibody that's actually designed for this specific CD276 protein, and then antibodies are linked to a warhead, which is like a chemotherapy payload, which then is released when the antibody hits that target. So this was actually quite a relatively new design, right? And we haven't seen this before for CD276. And then what's really exciting was that several months later, so Merck, a large company, sort of then independently actually designed the same antibody drug conjugate for CD276, and then they experimentally tested this in human populations to show that it's actually quite effective. which received FDA breakthrough designation. So this to us was also really exciting as a way to sort of independently see how the designs from a virtual biotech can then be independently corroborated by human studies, in this case from Merck. In order to design and make all these thousands of agents work effectively in science, we found that that's actually really useful to actually create a new layer of infrastructures and environments for these scientist agents. So for example, a lot of existing scientific databases, right, including databases that contains all of these biological proteins, they're mostly designed to be consumed by humans, or also consumed by pre-AI kind of algorithms. So often these databases and APIs are too brittle and too limited to be used by agents efficiently. So what we did was actually then take all of these existing databases, right, that contains millions of proteins and molecules and gene expressions, and then convert all of that into agent-native virtual file systems, which makes it much easier for AI scientist agents then to be able to synthesize and then aggregate information across all of these different data sources. And here's just a comparison that shows, so we call this paperclip, and then here's a comparison that shows that if you actually use this kind of agent-native virtual file systems to represent all of the scientific data sets and knowledge, right? So you actually get much better accuracy compared to, let's say, if you use Claude Science for the latest Claude models, right? But it's also a order of magnitude faster and cheaper than if you give the same language models access to these databases through the traditional MCPs or APIs. So we've actually gone through this process of essentially using these virtual file systems now to agentify all of scientific knowledge, which includes all of the papers that people have published, full text papers on the archive and other places, and all these different databases. That's all actually under paperclip. It's all free, actually, so you can then, with one line of code, then give all of that knowledge to your agents in this virtual file system that enables the agents to be actually much more effective in leveraging this. So this is something that one of the infrastructures that we built that enables the virtual biotech to be much more effective. So the last part I want to mention is now, we talked about how we can go from a virtual lab with tens of agents to a virtual biotech now with tens of thousands of agents. But what happens, can we scale that even further where we have millions of scientist agents that collaborate? So I think this is sort of a really exciting opportunity for us to really reimagine what all of human knowledge would look like when we should identify them. Right, so the past hundreds of years, the way that humans represent knowledge is really in the form of these very passive artifacts that we call static papers, right? So people will write this paper, but often it's very difficult for readers or for collaborators to figure out how to adapt the knowledge from that paper just by reading the static words. So our idea is that why don't we turn all of those static artifacts of knowledge from papers into dynamic and interactive virtual authors that we call paper agents, right? So each paper agent then is like a virtual author that represents all of the knowledge from that paper. So in this case, right, so here I can just ask the paper agent, right, can you help me to apply the method from that paper to my own data sets, to my own problem, and the paper agent then will do that automatically and generate reproducible workflows and come back to me with the end discoveries and results. So the goal of this is that by identifying all of these passive knowledge into dynamic virtual authors, then this makes knowledge much easier to disseminate, also easier to form collaborations. So under the hood, right, we created actually this paper-to-agent automated workflow that converts static knowledge into dynamic agents. So briefly, what happens is that we have a bunch of different worker agents that goes into the original paper and any associated code and data. And these virtual agents, these worker agents basically creates a virtual environment to try to replicate the original research process, for example, by replicating the results from that paper. In the process of doing that, then we actually create this paper agent, like a paper MCP, which then can be turned into a paper agent. So one of the applications of this is that now we can actually ask these knowledge agents to help to make sure the knowledge itself is actually reproducible. Right? So in this case, the paper agent is able to generate all the intermediate figures and results that matches the original publication. So that in this case, the paper was reproducible. But in many other cases, we also found that the paper agent actually identified key mistakes and limitations in the original publications. So the last part, the thing I'll mention is that I think maybe the most exciting application of all of these paper agents is that I think this enables a new form of collaborations between different agents, right? So we are now in the process of identifying every paper that people have ever published, so millions, that will create actually millions of paper agents. And now these agents are starting to talk to each other to generate new collaborations. So this is a real example of this, where Google DeepMind published a paper called Alpha Genome a few months ago. So it's paper to agent that was identified into an agent that knows how to use that tool. And then a separate research group published a genetic data set for mutations linked to GWAS for risk for ADHD that was also turned into a paper agent that knows how to use that data set. In this case, those two agents actually started talking to each other, decided they could collaborate, and then automatically discover a new mutation that increases the risk for ADHD, which is not known before. So we're starting to see many more of these examples of these paper agents starting to collaborate. So I'll wrap up here. So I hope to show these few examples to illustrate why we're really excited about this new axis of capabilities that emerges when we're scaling up the number of scientist agents that starts to collaborate and work with each other and also work with human researchers. Thank you very much.